Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. Shane Hegarty is the Chief Scientific Officer of Axonis Therapeutics, a Boston-based biotech targeting KCC2 to revive neurons and treat people suffering with neurological disorders. Shane shared with us the story of his transition from academic to biotech entrepreneur, why communication, persistence, collaboration and focus are critical skills for success, his amazing track record of securing grant funding, and much more. Well, we're back on Careers in Discovery with Shane Hegarty of Axonis Therapeutics. Shane, welcome to the show.
Absolutely pleasure to be here, albeit virtually, but great to be on the podcast. I've been listening to a number of episodes. Very, very, informative and good to hear other people's stories and see where the parallels are. And
Yeah,
I think it's a great platform. And I was hired to be invited on today to tell you the story of Axonis and myself.
thank you. And we're delighted to have you. And we always like to start a little bit with what you're doing now, as you may know from previous episodes. So I'm really interested to find out more about KCC2, about Axonis, and about the work that you're doing there.
Definitely. Well, Axonis Therapeutics is a company that we spun out of Boston Children's Hospital and Harvard Medical School. It's operational since about 2019. I'm the Chief Scientific Officer, co-founder of the company, and we're developing first-in-class and best-in-class oral medicines targeting KCC2 for a range of different neurological disorders, primarily focused on epilepsy, pain, and spinal cord injury in the near term, but we're very excited about therapeutic potential for KCC2. We're making the first drug to target this clinic. Hopefully, at the end of the year, we can start clinical trials. So it's all it's a great stage to be catching up with yourself and telling you what we've done to date and to put us in a position to be ready to do clinical trials.
Yeah, absolutely. And I appreciate some of this might be proprietary, so please don't worry if you can't say. But
Sure.
why KCC2? And tell us a bit about the concept.
So we almost stumbled across KCC2 as a target without much of a hypothesis-driven, literature-based approach. When I was lucky enough to be a research fellow in Boston Children's and Harvard, and at that institution, Professor Zhigang He, who's a world-leading neuroscientist, has a background in genetics, and he was in favor of doing, number one, in vivo research, which was a new experience for me at that level, and also I was in favor of doing unbiased phenotypic forward genetic screens, where he would ask a biological question and let the data drive what's the most important discovery. And what I really was enamored with the approach was that we would compare a lot of things side by side at once. You'd have your standard approaches included in these screens, and you'd look at whether any new innovations were demonstrating superior efficacy or phenotypes or some sort of superior property. And in that process, how the lab discovered KC2. So when I joined Gigant's lab, this screening project had already started. And they set up the screen to ask a question about spinal cord injury, where patients have incomplete injuries. So if you look at a functional MRI, you'll see spare tissue bridges, but this tissue is functionally dormant. So they have complete paralysis despite the spare tissue. And what had been reported in clinical case studies is that you can reactivate it with stimulation devices, epidural stimulation devices. Quite complicated to fine tune the location of the electrodes and the settings to get it to work, but sometimes with great benefit. And Zhigang was very excited about this breakthrough. We wanted to understand what was the biological basis of this. And functional restoration. And it was a neuromodulatory device that was achieving this. So he screened many different neuromodulatory compounds for the ability to reactivate the dormant but spare tissue left in an injured spinal cord. And so he took many different, we'll call them tool compounds, some of them on the market, targeting different ion channels, transporters, anything neuromodulatory in nature, things that when you deliver to the mice peripherally, it would get into the CNS. And that was the
Okay.
selection of about a dozen different compounds. So quite a labor-intensive screen. We're talking, you need many animals to model this disorder, but it was worth it because what we found was that only the KC2 tool compound was able to reactivate the spare tissue, and basically the mice could walk again. So it was quite a severe model where there was complete paralysis, no spontaneous recovery, and only the KC2 tool compound restore a stepping ability quite consistently and chronically in this model. And there was a lot of follow-up studies with genetic approaches and figuring out with cells and demonstrating that was indeed KC2 mediating this benefit. And that was the breakthrough discovery that led us to want to translate this preclinical research into a new therapeutic approach, initially for spinal injury.
Yeah, fantastic. And it sounds like this idea of sort of reviving the neural tissue has wider implications, of course. You said you're looking at epilepsy now and at pain and potentially other indications in the future.
So where KC2 fits in is it's the chief CNS-specific chloride transporter, and it has a vital role throughout life to continue to transport chloride out of neurons. And that sets what's known as the chloride gradient. At that point, you're trying to figure out where is that important, but it actually is the mediator of inhibition in the nervous system.
Okay.
So a lot of people are very familiar with GABA and it being an inhibitory neurotransmitter, but GABA A receptors are just ion channels. They open up and allow chloride to flow based on the gradient. If chloride flows into a cell, you get hyperpolarization and inhibition. If chloride flows out of the cell, you get the opposite, depolarization and even excitation. And early in even in human development, first 2 years of life, GABA is actually excitatory. And as KC2 expression increases with brain maturation, you get a switch from GABA from being excitatory to inhibitory. That's the first critical role that KC2 has during development. But during adulthood, it needs to continue to function to maintain the chloride gradient that the neurons will respond to GABA being inhibited. And there's a whole host of neuropathologies that impact KC2 function at synapses so that GABA no longer is inhibitory. And it leads to disinhibition of different brain circuits depending on which are affected. And that can lead to a lot of pathological problems.
Yes.
And that's what we're now aiming to address with our first-in-class medications targeting KC2.
Yeah. Fantastic. And so you were working on this at the Boston Children's Hospital and Harvard, and obviously a lot of excitement about it in the lab. I'm always interested, at what point did you and the rest of the team think, you know what, actually there's something here, we should go and do something with this, we should spin this out and take this on the next step?
Yeah, it's a great question. And when you're in a lab like this, world leading lab, Nature and Cell papers quite regularly, and you have all these different discoveries still in the lab, somebody's following up with a previous breakthrough, looking at new applications, combining different things. There was a lot of exciting research in the lab. Axon regeneration, Hegarty was the first to demonstrate a cell autonomous mechanism that could help CNS neurons to regenerate, and a lot of interesting neuroprotective research, interesting things around remyelination and inflammatory problems. But this was the approach that had the greatest functional recovery we had ever seen. So it was beyond kind of dramatic, even anatomical phenotypes that were, still used today as a positive control in different fields. There was a clear functional improvement, and that was kind of what set it apart from anything else that had been discovered. Just just look at these videos of these mice walking again, and then look at all the other things that have tried and had not worked, And we clearly were onto something very exciting. And, in the process of spinning it out, the company had existed because many different people were excited about G-gang A's research. And he had a lot of discoveries I just mentioned, axon regeneration being one of them. But Joanna, the CEO, and her other co-founder, Corey Goodman, who was, number one, a very esteemed neuroscientist. He won the highest prize you can win in the field called the Google Neuroscience Prize for characterizing different guidance cues for axons. But in the last 20, 30 years, he's built a number of biotech companies with successful multiple drugs on the market. And now he's managing them bio. So we managed to benefit from his neuroscience knowledge and his, being a world-leading biotech veteran. And so he was excited right from the beginning by Zhigang's research. And it was actually Zhigang and Joanna, when they were thinking about the different program, we had multiple at the start, right? We were interested in axon regeneration. We were interested in neuroprotection, all coming from different in vivo phenotypic screens we had done. One that I led with Joanna, which was we tested thousands of genes one by one in vivo using AB and CRISPR technology and functionally finding therapeutic targets that way as well. When we really thought about it in terms of what's ready for translation, it was this KC2 neuromodulatory approach. Kind of because what I just said, everyone knows that GABAergic inhibition is a validated therapeutic approach. People are using benzodiazepines, and there's other GABA PAMs, GABA receptor PAMs, and so that's a validated therapeutic approach. But the problem is some patients don't respond, or they can only take the drugs for a short while because of tolerability issues or other limitations. And what we were excited about was fine-tuning this inhibitory mechanism for prolonged benefit. But also a key differentiator would be, because we're not globally depressing brain activity by acting on an ion channel, just hyperpolarizing all the neurons at once, we are actually fine-tuning inhibition to restore physiological GABAergic mediated. That's the big difference. So we're leveraging the historic translational success of GABA mediated inhibition, but we're doing it in a more precise fine-tuning way.
Right.
That should for much more tolerability, but also address drug resistance that we see in epilepsy and in pain particularly, where even though there's been, especially in epilepsy, maybe 20 different anti-seizure medications approved, all helping patients to this day, the number of drug-resistant patients remain. And it's because they're acting on similar mechanisms. So this novel mechanism may address that, and we've got data to make us excited about that possibility.
Yeah.
These are the types of clues that this is science ready for translation into the clinic. Because, as I said, there's a lot of clinical validation. And the other fact was that there were patients who had KC2 mutations, had epilepsy. So they had the early onset epilepsy, autism spectrum disorder, a lot of other behavioral problems. And there was also larger GWAS studies associating KC2 mutations with a number of different psychiatric conditions. So it was a genetically validated target.
Yes.
And the final point was KC2 is selectively expressed in the central nervous system. So that made it a very attractive target for small molecule.
Yeah.
We didn't need to worry about KC2 in other organ systems. How would we, maybe there'd be some on-target issues in other organs that would limit the ability to drug KC2 in the CNS. We didn't have to worry about that. So that was all that together made us very confident that this was the most mature science ready for translation, just as long as we could make the first in class.
And that's the key thing, right? You got to get there first as well. But there's lots of science there saying you should go ahead and do this.
Exactly. We're benefiting from maybe a decade or two of world-leading science from many different labs. The first report of KC2 being important was about 20 years ago by the chair of our scientific advisory board, Yves DeConnick, and they discovered that in neuropathic pain, in the dorsal horn, in the spinal cord, there's a mechanism that should be suppressing painful signals, a sensory gate, and that's failing. And that it's failing because of KC2 hyperfunction. the inhibitory neurons there should be suppressing these signals so they're not painful. And without KC2, the inhibitory neurons aren't able to do that. And you get a facilitation of pain, process known as CNS sensitization. And so that was his breakthrough. And he's been working on it for 2 decades. A lot of initially in pain, but now in many other indications. So we benefited from these a lot of different labs getting very excited about the target. And, because it's got such a critical homeostatic role in the nervous system throughout the lifespan. Whenever someone really decides they want to study it in their model system, in their indication, they tend to find an important role for SC2, and that, opens up therapeutic potential for us. Then it makes it more difficult in terms of indication prioritization. There's a lot of different ways you could go and test the molecule, and we have a lot of exciting discussions and even inbound requests around where we could start to look at the effects of these drugs. But we have a lot of time thinking about them, forms our research and our R&D efforts.
Yeah, absolutely. And it's just as important deciding what not to do as what to do. And I want to come back to kind of the formation of the business, the growth of the business. And you've mentioned some of the amazing people who are involved in the project. But I want to talk about you a little bit first, Shane. And we always like to go right back to the beginning. And I'm very interested in the origins of people's careers. And so, take us all the way back to you being there in Cork. Why science? Why neuroscience? Why this path for you in the first place?
It's a great question, and there wasn't a strategy to it at all. And what I did know is that I didn't want to work in the local chipper or the chip shop anymore, which I started doing at the age of 15. And then what I realized was from that experience and from experience of my wider family that not having a specialized skill set was going to make it difficult in terms of doing something. And luckily, I was good at school, and that really had options going into university. But even then, I couldn't decide what I liked the most. I was thinking pharmacy, I had accounting up there. I was thinking about science, and it was difficult to really what I wanted to do. So I picked quite a broad science course, and again, doing quite well. But it took me till about my second year, still getting different scholarships and things like that. But for when it really picked up, now I've got this passion area, and it doesn't feel like I'm studying anymore. I just want to read this and study and, dig into it deeper out of pure curiosity. I read this book when I was traveling, and by Jill Bolt Taylor called My Stroke of Insight. And it was this neuroscientist who had a stroke. And in the book, she first had to explain what the brain does, and then spoke about her experience of having a stroke and how she understood what was going on and how she recovered from that kind of background expertise in neuroscience. So I became fascinated over that summer about the function, plasticity, and potential of the human brain. And luckily, in third year at UCC, you could study neuroscience as a subject, without much exposure to it before that. But after reading that book, I knew that this was the area I was fascinated by. I just didn't know enough about neuroscience, the human brain, even psychology, which my mother has begun to study as well. I have a lot of interesting books around that house. So I knew that this was where, I could study something and feel like it was a pleasure, and which is kind of a weird thing to say because it took me a while to get to that point. And what that meant was I could spend a lot of time researching and reading about this area. And it was then helping me be, maybe at the end of the undergraduate top of my class, that gave me a lot of options to first be in a position to get, stipends and different PhD opportunities, but at the same time, I had the opportunity to go to the lab in UCC with Aideen and Jeremiah that ended up being my professors overseeing my PhD, and do a project that I was very interested in. And that went well, and we could propose a independent PhD project that was building on some of that undergraduate work, and that was funded as well. So it all came from this kind of, I got getting really passionate about an area and just digging into it and just enjoying the process of doing it without really knowing where is this leading to. I didn't have There was no one that had done postgraduate, education in either side of my family before that. There was no, major career guidance going on. It was just Shane loves neuroscience and he could spend all day, reading about it, researching it. And it seems to be working out well for him. He's getting to do this PhD. It was that type of process. But it was very rewarding when, as I dug into it and I had these fantastic teachers and mentors who were patient with me, were patient with my curiosity, all the questions. I wanted to know things that we didn't know yet about the human brain and its pathologies. And they were encouraging me to dig deeper and
Yeah.
not dismissive in any way. And, that helped me to just go and kind of test out these ideas or hypotheses that I had, or, already starting to write maybe a review article about a whole area, even though I know maybe credibility in neuroscience as a young PhD student, because I just wanted to know the state of the art for myself. Also kind of from a position of imposter syndrome. I'm in this new area. I don't know everything. Why would anyone listen to me? I'm going to have to read the whole fields, research papers, write my own review. And then I'm doing that as an exercise for my own understanding. And then it kind of put me in a position to have a knowledge base that I could build on, take the next step in terms of innovation or research. And that was my process academically. For
Yeah, I
many years.
see. So, and it's interesting, isn't it? Because I suppose as you develop as a scientist, you're almost deliberately putting yourself in positions of ignorance, right? Because you have to go and explore things that nobody knows anything about.
Exactly.
There's context and there's surrounding information, but going to those places where there is no knowledge is kind of the point, right?
Exactly. And if you have people around you who kind of trust and empower you to do it, rather than are too prescriptive or too using you as like a pair of hands or something, just go test my idea. I never had that. And when I was coming up, it was very self-directed. I was getting these scholarships and fellowships on my own proposals that I co-developed with my PIs. So I felt like I was driving the research direction myself in areas that I was really excited about. So I could spend a lot of time on it. Rather than maybe if I was assigned or just go and do this project on something, maybe I wouldn't have had the same level of commitment and excitement about it. And, enthusiasm isn't, enough to sustain a whole PhD. You have to be, that's a great starting point. You have to actually somewhat enjoy the process of failing and figuring out why things are not working and tuning things to get to a point where you've got a new level of understanding or something is working and now you've got a way of pushing kind of, the research forward.
Yes.
And that was very productive for me for many years. And that kind of led me through, PhD postdocs were these passion projects. And I had opportunities to go elsewhere, different postdocs or PhDs elsewhere. But it was just the fact that it felt like it was my own research kind of direction. I didn't want to kind of give up on it. I needed to get to the end of it kind of thing. I needed to, we started out the level of the ligands, figure out the receptors, figure out intracellular proteins were by the end were in microRNA, and just like total characterization of this area that I was really deeply interested in. And that was very productive for many years. And while I was there, I had the opportunity to take on a lot of teaching responsibilities and like do some courses. So as I was specializing down in this one area, I was getting at the same time an ability to broaden or even revisit things that I should have known from my undergraduate that you might get as you just so into something so niche in your research focus. So I was broadening my knowledge or revisiting things, and that was helping me thinking about problems from many different perspectives. And then what I felt like the key competence from that whole time was if I can, explain it simply to undergraduate students or whoever it is, then I don't understand it enough, or they're not going to be excited about it. And that's become so important now in my career as an entrepreneur, innovator, scientist, where I have to continually do that in a way that creates a sense of urgency or excitement about what we're doing, why it's meaningful, why it's ready, why this is the time to push this forward. And that's kind of really important. And also, as I mentioned, in Ireland, there wasn't a lot of funding around.
Right.
So you're kind of having to apply for funding almost every year to even have a job, kind of that basic level of survival, but also to keep your research going, right? You're excited about this. You know that you want to do all these extra steps to really understand it more deeply. And so you're working on these defined timelines, which ends up being like how you operate in biotech,
Yeah.
short kind of milestone driven projects. But also while that's happening, already thinking about how you're going to fund the next step, how you're already, preparing for that and being comfortable with the uncertainty of, you need to deliver on what you're doing right now, but already be preparing for the next step and communicating to everybody why this is important and why this is valuable. And be, that was, unbeknownst to myself at the time, as an academic, just wanting to be a professor, all those skills, all that experience has really set me in a good kind of base for, what I'm doing today, which is a biotech
Yes.
co-founder and entrepreneur.
Yeah, absolutely. And I think that communication of science is really key, isn't it? I think absolutely, as you say, in a fundraising context, but also from a teaching point of view. I always say to people now, I'm not a scientist. I find science fascinating now because I'm speaking to people like you who are doing these incredible things. The way it was communicated to me in school was very different. And I think,
Yeah.
there's a key thing there, isn't there, that you touched on there, that you've got to go to the right level to make it interesting to the people you're communicating with, to
Yeah.
stoke that passion.
Exactly. You need to understand what would engage the audience. What do they care about? What are you doing? Where's the bridge there? The concept of entry points, whatever you want to call it. There's all sorts of terminology or pedagogical jargon you could use to talk about it, but really is about engaging with someone, trying to make them excited about what you're doing, wanting to, why would they want to spend time learning more?
Yeah.
You gotta kind of always be ready to do that in multiple different stakeholders. And, I find that really enjoyable. And, I don't know if it's through practice or, natural ability that I find myself, quite good at that. Usually typing, with the Irish accent, maybe people can't even understand what I'm saying, depending on what country I'm in. But that's been a, process of adaptation, even moving to the US, trying to have people understand or slow down the way we talk. So it's all been fantastic experiences for me to continue to learn and grow in a way that hopefully will sustain a career in making new medicines for neurological disorders. It's all, none of this was pre-planned. I wasn't even aware of biotech as a thing, to be honest.
Right.
Before I became to Boston, I obviously knew about industry. There was maybe a old school thinking that I would be giving up on my own lab and things like that if I was thinking about that. And even when I was in Ireland, before I came to Boston Children's Hospital and Harvard Medical School, I was working on the faculty at my alma mater, but I had a chance to start my own lab in a tenure type position in another Irish university, which
Right.
was exciting. But I was in this scenario, continued specialization, kind of narrowing down, almost in a way I felt like I was pigeonholing myself. It's almost like I could never do anything else. So I wouldn't have the credibility to do something slightly different. And not that I wasn't that excited about what I was doing, but at the time, a couple of clinical trials were failing in this area, the neurotrophic factors for Parkinson's disease. And I couldn't ignore that. I did a systematic meta-analysis for myself to figure out maybe if there were multiple trial data together, we would see something. And that didn't, remain true. So I felt like, do I really want to spend the rest of my career on this area? And luckily, I had the opportunity at the same time to go to Gian's lab. And when I went into his lab and discussed the types of projects he was doing and what we were going to plan for when I was there, I couldn't turn that down. It was an opportunity too good to not do. This highly resourced environment, cutting edge research, the recent, that was a big difference to some of the stipends I had in Ireland were $5,000 for a year by a couple of suppliers. Here was think big. There was no financial limitations to what we could do.
Yeah.
And it was just anything, the more ambitious, the better, which was great. And that was, good for thinking big, but you still have to be focused.
Yes.
So it's like, even when you have all these options, we talked about basically having broad therapeutic relevance earlier. When you have all the options, you still have to focus. You still have to deliver on something of value, with some clear, milestones and deliverables in mind. So that was, all this is, you don't really know what you're going to get out of it as a learning experience before you get in. For me, it's normally I'm here to do a research project. I'm interested, and all these other skills are just benefiting along the way. Now, retrospectively, I can see why these different experiences have put me in a position to be able to build the company we have today.
Yeah, absolutely. And so did it feel like, with the greatest of respect to UCC, did it feel like a step up when you went out to Boston?
Well, look, it's an interesting question because when I got there, imposter syndrome was through the roof.
Right.
A lot of my friends were joking about goodwill hunting and things like that, and from Ireland. So there was a trepidation about Finn will be found out that he's not, a great neuroscientist. Because at that point, I felt very confident in my abilities. I'd been around Europe training as well. I went to, AU Live and the Erasmus MC, and I'd been meeting a lot of different individual conferences. I felt like, I'm one of the, up and coming, hopefully worthy neuroscientists. And I even got a prize in Ireland called the Neuroscience Ireland Investigator Award that kind of validated what I had achieved to date, even at quite a young age. So I was quite confident, but then, I was still worried. But when I got there, even in our own lab meetings or any departmental meeting, what I realized was because we didn't have a lot of money to do, huge experiments or, a lot of different things at once, almost distracting amount of things at once, we had to spend a lot of time reading papers,
Right.
a lot of times being very, hypothesis driven. We could really only test one thing. To test one thing, you need to be selective. You need to think very deeply about your plan. You can't waste anything. And not to say that there's waste going on, but the idea of just kind of testing everything, everything is open. Not that it's a bad approach, very broad, and it can catch a lot of different, when unknowns when you go about it that way. But I felt like on a theoretical level, I might have been in a better position.
Yeah. Okay.
No colleagues there, but that's what I felt when I was there. And it, came back to another thing where asking the right question. What's the most important unknown in terms of at the end of this question, you have something that's going to help be of value to anybody. For me, it's always, is this going to be helpful in the patient? Is there some sort of therapeutic benefit for doing this experiment on the back end? Is this leading to more, not just knowledge for knowledge's sake? I've always had kind of what I thought was translational research projects. And I didn't every paper, this is translational. And that was also starting to bother me as well. I never was able to figure that out. So the only way to figure that out was go and translate it breakthrough yourself through a biotech company, make the medicine, the definition of translation or translational changes as you go, but to actually go about and do it, that was a burning kind of desire of mine, rather than kind of hypothesizing and just leaving this open question out. This could be translational because we've done this set of experiments. That would become not enough for me, especially when I saw, almost this mind-blowing kind of discovery right before my eyes.
Yes.
That was kind of where we were. So, that's how I felt there. And it was great training in many ways, but at the outset, the training we had in Ireland was, at a kind of academic level, theoretical level, just knowledge level, neuroanatomy, whatever it is, that part is at the standard of any came across any Harvard Medical School in Boston, Children's Hospital.
Yeah, and it sounds like that sort of resourcefulness that you learned and the working in that resource constrained environment actually gives you really good habits to then take into a biotech company, right? Because again, everything costs so much and there's only so much you can do. You've got to do the right experiments, not just every experiment.
Yeah, the discipline and the focus.
Yeah,
and that's essential. And the opportunity cost of thinking you want to do something else. All that. It really is. It was by necessity before. Now it's by strategy and resourcefulness and wanting to move quickly on one defined goal, which for our company is test the first in class medicine in the clinical at the earliest possible. What would a drug that's high quality, right? A drug that is going to make it to market. Just that everybody, whoever would look at it, large pharma, whoever would be interested in it, they would stand behind it and say, yeah, this is best in class, not just for
Yeah.
the class.
Yeah, absolutely. And so, thinking about people who might be listening to this, who perhaps are in a postdoc environment, have discovered something that they think has real potential, are thinking
Yeah.
about taking that step out into spinning out a company. What have been over the last few years? What have been the things that you've learned that you'd share with them that you think have been the most important lessons in terms of going into that biotech environment?
Um, yeah, well, I think I benefited a lot by where I'm located, which is in Boston. I've got, this fantastic ecosystem around me where you can get started very quickly if you've got the right intellectual property idea. And there's a lot of kind of bridging program to become even educated in what building a biotech is all about.
Yeah.
When I was a research fellow, I was able to do the Harvard Innovation Labs venture program, President's Innovation Challenge. They're teaching you everything along the way. You've got these world-leading advisors volunteering their time and guiding you even at the early step. And they're building your confidence. They're telling you
Right.
that you've got something of value here, you've got something ready to, to go and build a company around. So you don't just, take a stab in the dark and all, you know, wish for the best. You have a lot of different people who've done this many times who are validating what you're, and they're also telling you what not to. As I said, when we started out, we had a platform technology. These are unbiased in vivo phenotypic screens that were very productive for us. We could have done that for different disorders as well. And we had a couple of different therapeutic targets we were really interested in. SC2 was one of them, but we had others. And with that type of, going out and getting battle testing your ideas, right? Go out and you have to tell people what you're doing. Obviously, not everything that you preserve your key IP. You have to go and talk to KOLs, VCs, different people in the industry, so they can challenge you and you either what you need to work on, get to the right level, or what not to do or what's not ready. So that's just, it's kind of, I've been like that from day one, kind of just putting myself out there
Yeah.
and almost taking these calculated risks of being willing to fail and learn from those mistakes as you go and iterate quickly, not be disheartened. Just be happy that you're one step closer to the right answer because you started.
Yeah, absolutely. And I suppose having that clarity and sense of direction is really key, right? So you can only be one step closer if you know which direction you're supposed to be stepping in, I suppose.
Exactly. And that, that, as I said, if you need to kind of understand the state of the art and find where there's actually meaningful kind of gap or meaningful improvement, a significant kind of step, because it has to be worth the effort. Like building a biotech is not easy, very difficult. You have to spend, almost every waking hour thinking about it, wanting to move forward. And so to do that, you need to be sure you've got the right, purpose. You need to be in the right place. For me, Boston, the IP ecosystem is perfect place to build a biotech.
Yeah.
And the people that you want to build a company together with, ideally, that's a group of people who share the same purpose, who you kind of synergize with, that you're helping each other. So different kind of expertise, viewpoints, kind of challenging each other to bring out the best result. Because, if I went down having my own lab, even in Ireland, I had the opportunity to operate, even continued through even the US system, you would be, you're a PI in your own lab, kind of almost in a little silo.
Right.
And you want to kind of carve out a new area for yourself and dig into your own, best kind of whatever you think is best. But when you've got a group of other, high performing individuals with different backgrounds and expertise, and you're challenging each other all the time in a healthy way to just get the best answer, not what Shane thinks is best, and we're always doing that. We're always willing to look at, what we have and whatever plan we're planning going forward and challenge it again, even if it's painful. That process of iteration, or what we call in the industry the pivoting ability. That's what's key for biotech. We make data driven decisions all the time based on what we learn or what is happening in the environment around us. So that's essential quality for success. And just, I said, having the purpose and the people around you that cannot help you to persist, because the persistence is key. Because it's not going to be easy. Because with anything in science, You were always testing new
Yeah.
directions, and you learn from that, and and then move forward. So that persistence is important, with as I said, the focus and the resourcefulness and all that. So and while all that is happening, building a great team that buy into the vision, and then constantly looking for funding from VCs, or in our case, we also have about $8 million in grants. So we was grants to try to, we've managed to raise about 34 million in VC and angel backed dilutive capital, but the grants have been really important because they generate momentum, they validate your science for everyone else. And when they see NIH and other people have bought in, foundations for building relationships with foundations around who can excited about the technology, and they're involved in it early, and that extends your runway, and it's great. That was something that there's some people, they might not like academia anymore because they hate applying for grants, but I hadn't got to that point yet, and I was able to bring that, almost, it's a difficult process, but it's still enjoyable. It helps you focus your thought as well. You got to articulate, a plan for a year or two, and you be ready for, again, putting the idea out there, let other people criticize it, and whatever comes back, you can improve. And rather than keeping it all to yourself or waiting until something is perfect, you work at promise,
Yeah, absolutely. So it sounds like all the way along, you've been really keen to, as you've said, expose your ideas to the world and see where the weak points are, see where the stress test them, as you say.
Yeah. Even at the risk of, embarrassing myself, that willing to, obviously, you want to have that confidence, that base level, do your homework before you go out. But at some point, there's a point at which, like the print, like 80-20%, principle, where you're 80% of the way there. And that last 20% actually might come from you picking your 80% fan and going and asking people about it. Just like let them, they give you feedback, criticize, whatever they want to do with it. And you don't have to, every time someone says something, doesn't mean you have to tear up what you did and stuff. You can listen to the consensus and start to, fine tune it. And that's kind of been really helpful all the way through as well.
Yeah. Or it might even be, I suppose, just one thing one person says that unlock the
exactly a new angle, a new perspective that we've always tried to do. I mean, this was something that surprised me about the biotech, being in the biotech, the ability to collaborate. So people in academia, there's an idea that collaboration is strong, but then sometimes there's competition. And obviously with private companies, there's competitors in terms of other companies. But for me to collaborate with, world-leading veterans in the biopharma industry who are now consultants, or any academic lab in the world, the ability to collaborate with them has never been greater, because they're excited to work on a new medicine about to go to the clinic. And we're able to bring in these different labs. Sometimes some of these labs might have not seen eye to eye even. SC2 were leading labs, and they both work with us, and now they're helping, and they're almost, cross-validating each other. They're both right kind of thing, and helping us to go to the next level of, getting over whatever hurdles there was in drug development for this target, the new target. We need customized assays. It's not easy. There's nothing off the shelf about doing research on SC2. And so that's been very, very rewarding. And you get a course in all these different areas, right? I'm a neuroscientist by training, spent a lot of time, obviously, preclinical neuroscience research, but now you need to learn about everything, all the way up to clinic, including how to make a drug or discover a drug and things like that. So what's been brilliant about, and it's almost like when I did a PhD and I was starting to teach, specialization combined with generalization.
Yeah.
In terms of knowledge, you're broadening your horizons in one area, and you're also specializing in one really important thing that's differentiating you from everybody else. You kind of have both things going on in parallel, and they almost help each other, because it's like, take a break from your specialty and you open your mind to other things, and you go back and you kind of get a better way of progressing your specialty, let's say. And what I loved about, and what I'm still loving today in biotech, is learning all these different functions. As I mentioned, regulatory, chemical, commercialization, drug development. Not that I would, you were relying on experts in these given domains to oversee get the given function, but you have to be able to communicate with these people, learn their language, understand the basic background of the different disciplines. And that sounds like, if I was to do courses and all these things, it might take a very long time. But by almost running a drug development program into the clinics, you learn it all as you go, and you kind of, with the experts kind of guiding you all the way through. And it works both ways. You kind of want to get the best out of them, and by kind of pushing them to think a little bit more deeply about whatever guidance or advice they give you. Just, even if you ask me now in something, I'll give you the first idea in my head. It might be quite good. It might be nearly there.
Yeah.
But if we keep talking about it, I'll give you a better answer. If you kind of just push me a little bit more in a kind of healthy kind of pushing then away. And we like to do that in every part of our organizations, or even someone working a day or 2 a week as a consultant somewhere. And, that's been really productive to drive excellence, I think, in areas.
Yeah, 100%. I think, in any business, one of the best things you can do is go, okay, good. Is it the best we can do?
Yeah, exactly. And that's kind of like, you could spend forever trying to get something just marginally better.
Yeah.
But at every point, there's always the point where you can improve the quality in somewhere.
Yes.
And just be willing to, go through the extra effort of pushing it just a little bit further, and then knowing when, okay, this is the best we have, given the timeline and what the next step will be. And so I think there's a balance there, but
yeah,
it's good. It's good not just check there. Let's say at the very minimum, don't take the first idea with every head or every different step of your journey.
No, absolutely. And I want to touch on this. I don't want to spend too long on it because it could be a very long conversation. But I think the point you make about grants is really important. Because $8 million isn't going to get you there, but it's not an insignificant amount of money. And
exactly.
I think grant funding is really neglected by a lot of biotech companies.
Yeah.
But that's been a sort of deliberate part of your strategy, it sounds.
Exactly. It's something like, we have that routine or habit of, from academia anyway, not missing any important calls.
Right.
How would you miss? So it made sense. We are doing this research anyway. We know what we want to do. It's exciting. Clearly, it's breakthrough research is, recently published in the best papers is about to translate, huge potential. So why wouldn't this be a great grant? Of course it is. So as you go, you write these different grants, and for the other purpose we use them for is something that's not one of the programs that maybe isn't, we're using our capital for. We can call it a side project, something like that. And we know it's interesting. We know it's important, but we're not going to dedicate capital right now because we're very focused on the one goal. But why not have this other parallel track of research where we're collaborating with an additional world-leading lab, and they're doing, not that we're bringing all this work in-house, but we're collaborating on this important area that only they can do really well. They're excited about it, and we'll write a grant together, and we'll have this data for maybe, a secondary indication, secondary asset, whatever that might be. So it's been a great way of like expanding or kind of horizons as well. And as I said, I think it validates our work. So when people are doing due diligence, they see these different, grant bodies, DOD, NIH, whatever it is, are brought into what we're doing, and it kind of de-risks it for them from that perspective.
100%. And it's not just about the money, it's about the mentality, but it's also about the fact that not only are you getting non-dilutive funding, but you're also making the core of your business more investable for those who want to
exactly
in equity fund.
There's of course a balance to it, grant cycles can be, protracted, and, you're waiting. So you don't want to be waiting, you don't want to be holding up the whole show waiting on the outcome of a grant. But there's a way to do it, and it would never slow you down, but it would only always be added to your overall goal.
Yeah, absolutely. So coming back to today and coming back to Axonis and the work you're doing, so it looks like a really exciting time, getting closer and closer to the clinic. Tell us a bit about, what's next, what lies ahead.
So at the end of the year, we'll start clinical trials. So we're very excited about that. It's healthy volunteers, just proving it's safe and tolerable. Like we're very confident about from any of the work we've done today preclinically. And then that sets us up to run trials later in epilepsy and pain, which again, we've done a lot of research to support those trials. And so that's kind of the plans for our lead molecule, first in class oral drug for KCC2. And we're also at the moment fundraising, and so that's going to support these phase 2 trials this year and beyond. And and we're always, you could say people are always fundraising. It's definitely true.
Yeah.
No, it's very targeted. We're ready to push ourselves into being a clinical stage company, and a great time to get that support and buy-in from other investors. And when you find the right investor, they become top partners. They're very experienced in these areas, and they really help and drive the company forward. And that's really, our executive chairman, Corey Goodman's like that from day one, and with that experience and that also backing us and passion for what we're doing, it's been the big benefit for us, and we want more people like that come in and help us in the next steps in terms of getting this medicine patients that need it.
Yeah, absolutely. Well, it's incredibly exciting. We have our fingers crossed for you. Thank you so much for taking the time, Shane.
Absolute pleasure. And I'd say we could talk for hours, the two of us. It's then to connect today. And I appreciate your interest in highlighting in what Axonis are doing, and giving me the chance to share some of my learnings over the last 15 years of being a neuroscientist. And I hope somebody can get any of the thoughts I shared, and can get inspired to want to make medicines themselves. And if anyone is interested in learning more, I'm very happy to catch up with anybody. I'm sure my profile might be linked somewhere. Happy to meet anybody thinking about, they have passion projects in research, and they really want to figure out where they can translate it to medicine.
No, well, it's a really kind offer, Shane, and it's been an absolute pleasure. Thank you.
Thanks for listening. If you enjoyed the episode, please like and subscribe, and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.